So I want to tell you about an approach that we're taking that we're really excited about because we think a lot of people who don't normally do single molecule work could approach this tool. And so what I want to talk about today is done by an amazing team shown here. I'm going to feature mostly today the work of Matt Shai. So I'm going to tell you about how we can watch fluorescently tagged proteins from nuclear extracts on the C-trap. And we've named this approach SMADNE. And I'm going to pick one example to show you the power of this tool. So this is basic scission repair. I'm from the University of Pittsburgh, where we have a very strong genome stability program. And there are 6 different repair pathways in mammalian cells. Basic scission repair is a process in which an army of glycosylases, there are 11 in mammalian cells, find base damage. And this particular base damage is 8-oxoguanine. It's about 1 8-oxoguanine every million bases. The steady state level is about 3,000 or 4,000 in each cell, and it's removed by this process shown here. So OGG1, 8-oxoguanine glycosylase, removes the damaged base, generating a basic site, which is then nicked by AP endonuclease. This enzyme, poly-ADP ribose polymerase, finds the nick and activates a ADP ribosylation step using NAD to make long PAR chains. That's believed to then attract XRCC1 and pol beta to fill in the gap that's generated. And then finally, it's ligated closed. This model is based on a large amount of cell and biochemical data, but I've told the guys in my lab before I retire or expire, I want to watch this process in real time and be able to understand how long does each protein sit? Do you ever see colocalization as predicted, for example? And is this pathway actually correct? I don't have time today to tell you about it, but we know that ligase, by using this tool, binds better to NICs than XRCC1. So we're thinking that this model isn't quite correct. But I want to focus today on what Matt has done on PARP, poly-ADP ribose polymerase. It's activated by single-strand breaks and double-strand breaks. It's actually important in all DNA transactions, transcription, replication, and repair. And as many of you know, PARP inhibitors are used to treat BRCA1 and BRCA2 deficient ovarian and breast cancers. So it's an important target to study. I love this quotation by Sidney Brenner. Progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And so my lab, the guys in my lab are pretty good at purifying proteins, but if we want to march through this repair pathway, we were thinking we need another approach that might go faster and be easier to do. So we decided to use nuclear extracts for 2 reasons. One, you don't have to purify the proteins. You can generate variants very quickly. But also, we wanted to begin to look at other proteins that you would get in a nuclear extract and see how that would affect the events that you're studying. So SMADNE is actually a play on words. If you're ever in Slovakia, this is a pretty good beer. It stands for a thirsty monk, and we're all thirsty for knowledge. So you can follow your favorite fluorescently tagged protein. You can watch it bind to DNA targets. If you have multiple colors, you can actually watch the coming and goings of multiple proteins. What we love is we can rapidly just mutate the plasmid and then express variants. We can look at post-translational modification. And a bonus that we didn't expect, but looking back, we should have. When we did mass spec on our nuclear extracts, the two of the top 10 proteins were heat shock protein 90 and heat shock protein 70, which keep your proteins happy for hours at room temperature. So this is the workflow. We get a plasmid or make a plasmid that has a tag on a protein of interest. Had a wonderful undergrad, grew up over 100 plasmids while he was working. We then take the plasmid and transfect 4 million U2OS cells. Nothing special about U2OS cells except they transfect really well and they grow like weeds. From 4 million cells, we get about 100 microliters of nuclear extract. We freeze 10 microliter aliquots. They're stable at -80 for a year. We take one aliquot, dilute it 1 to 10, and then put it on the LUMICKS C-trap. Now, I know most of you are familiar with the LUMICKS C-trap. I just wanted to show you. Nate was working with Jennifer this summer, and in 2 weeks, he was able to be driving the C-trap. So it works really well. As you know, it's made up of 3 very important components: optical traps, 3-color confocal, and the wonderful microfluidics. So this is a typical workflow. We capture the beads. We then capture usually lambda DNA. In this case, it's lambda DNA that has 8 observable nicks. Then we go to the buffer channel and wash away the extra DNA. And in this example, we use YFP tagged PARP, but we've used halo tagged PARP, works actually much better. We generate a chemograph, and then we can actually look at the on and off rates. So what kind of information can you get? So we use a nickase, and as I said, there's 8 observable nicks in lambda, which is 48 kb. So you can get the dwell times, that gives you an idea of the off rate, the gap times, the on rates, and you can see the binding positions. Is it only binding to the nicks? And if it was moving, you could get diffusion constants. So Matt really loves this approach. Here's a typical chemograph. You can see it's sampling many of the nicks over time. So the dwell times, we use a cumulative residence time distribution for each dwell time, and then fit an exponential. And from that, we can get the dwell time, then the off rate. And from the gap times, we can get the on rate. And we can actually use calibrated standards to use the background fluorescence to get an idea exactly what the concentration of our protein is in the flow cell. And so from the apparent on rate and the concentration, we can get a true on rate. And what we were kind of surprised is that it looks like it's diffusion limited. So that suggests every time that PARP comes out of solution, hits the DNA, it's a productive binding to the nick, which I didn't expect to see. So from the off rate divided by the on rate, we could, for the first time, measure PARP binding to a nick at the single molecule level, in which you don't have any ends. And most people who use oligos, you have to worry about end binding as well. So this is really exciting. We wanted to then do structure and function, and so we used AlphaFold to superimpose on a NMR structure of the 2 first zinc fingers that bind to nicks, and Matt generated this model. And it actually really looks good interacting with the DNA. You can see the zinc fingers and the WGR domain kind of wedge the DNA. And then in the backside is the ART domain. That's the domain that binds NAD and is active. And you can see that it's a long way from the DNA. And so one of the puzzles in PARP activity is how does it know that it's actually bound to a nick, and then how does it activate the NAD site? So the first question was, the literature suggested that zinc fingers 1 and 2 were necessary and sufficient to bind to nicks, and so we expressed just zinc fingers 1 and 2 with a halo tag on the N-terminus end. The Janella floors are really good. We do put Trolox in our system, which allows the photostability for well over 2 minutes. And so you can see that the zinc finger is binding and rebinding very fast. But again, the KD is almost as tight as the full length. So we think that most of the binding energy is through the zinc fingers 1 and 2. So we were a little worried that maybe we're activating PARP when we break open the cells, and it auto-PARylates itself. And so Matt made a triple mutant, and we said if we see differences, we can be assured that it's not getting PARylated. So he made the triple mutant, and we asked, does it change its binding? And we were really, I'll show you where the triple mutant is. It's back in the, where NAD is bound, and so this enzyme is catalytically dead. It was really shocking the first day when we ran this on the C-trap. It's so long-lived, it was amazing. So we had to fit a double exponential, and the average time was about 60 seconds. So it's actually increased the binding energy tremendously. And so we believe there's an allosteric, reverse allosteric network that the ART domain is now talking back to the WGR domain and the zinc fingers to make it bind tighter. There are many inhibitors of PARP, and we've started a survey. The one I'm going to tell you about today is a biochemical inhibitor. It's cell impermeable. You can't use it in cells. But John Pascoe has used it to suggest that it's a pro-retention. It makes PARP bind longer on the DNA. So we wanted to measure that. There's also clinically active ones that are neutral or mild pro-retention. And then there's a third class that are supposed to boot it off the DNA. And so actually this month, Jennifer is screening all of these. So EB47, they've crystallized it in the NAD pocket. It's shown here. And it interacts with this helical domain, and that helical domain then talks to the WGR domain, and John Pascoe suggested that EB47 through reverse allostery will be pro-retention, it'll make PARP bind longer. So we wanted to measure how much longer, and we were stunned. We could measure a KD now of 20 picomolar. That's 105 times tighter than the wild type that's not inhibited. So to summarize this part, PARP binds to NICS with an affinity of about 2 nanomolar. I didn't get to show you, but if you pull on the DNA, actually PARP binds better to NICS. Zinc fingers 1 and 2 are necessary and sufficient. This triple mutant now binds tighter, and then EB47 increased the binding affinity tremendously. Olaparib had no effect in our experiments when we first screened it, but we're going to go back and look at it and more. Well, ultimately, repair in the nucleus takes place with nucleosomes. And so as a first step, I was able to collaborate with Brett Freudenthal and Tyler Weaver, and using the LUMICKS arms, which Marco helped us with tremendously, we could then ligate a 601 nucleosome into the arms and then string up one nucleosome. So the Psi3 is on the H2A, so we know that the nucleosome is there. And then we have the fiducial markers with the autodie. So when we string it up, we can see one nucleosome in the center. So we also introduced NICS into the nucleosome. And so here we've nicked the nucleosome at just the dyad, and you can see that PARP, this is YFP PARP in this case, it's blue in the instrument. You can see that it eventually the Psi3 photo bleaches, but it bound directly to the nucleosome and nowhere else. And from that, we could get on and off rates. And from that, we could get a KD. And so it actually binds to a NIC in a nucleosome with almost the same affinity as naked DNA. The big surprise, though, so this is a movie I really like. This is YFP PARP. And I said that the DNA is wedged between the zinc fingers and the WGR domain. And this was animation that was done by Janet Iwasa at the University of Utah. And so when it binds, and this is actually a real chemograph that she's adapted. You can see the blue, and then it bound again. So from the gap time and the dwell time, as I said, you can get the KD. And so Matt really worked hard on this. And we were kind of shocked that the naked DNA and all the nicked DNA and even the nucleosome, the dwell times are almost identical. But that's only half of the equation. You have to look at the on rates. And the on rates actually differed. And that actually led to differences in the KDs. So an undamaged nucleosome is bound very well by PARP. It's been suggested that it can bind to nucleosomes, but all those experiments use a nucleosome that's formed with 2 double-strand breaks. In this case, we have the nucleosome buried in a long piece of DNA. And you can see that an unligatable nick is seen best, followed by the dyad. We don't believe that the protein is actually able to displace the nucleosome. We think it's actually binding through the zinc fingers. Bustamani's lab says when you pull with a force of around 10 piconewtons, you begin to unwrap this right arm here. And we were able to do that on the C-trap, and we were really shocked by what we saw. Now PARP binds to the nucleosome 10-fold higher affinity. So that would suggest that PARP in the cell is binding to chromatin almost all the time. Some people have done single particle tracking, and it doesn't look like it's doing that. So we think it's getting autoparylated and then deparylated. So it's coming on and off the chromatin, and that's something we want to look at in cells. The other interesting feature of these data is that you can see that when you put a nick in the nucleosome, the affinity, if anything, goes down. And so we think that that would suggest that it's primed and ready to do its work because it's already on the chromatin. So to summarize this part, at low tension, we saw nick DNA duplex is found with the highest affinity. Binding to undamaged nucleosome increases with force. In fact, all the binding entities seem to go up and the KDs come down when you add a little bit of tension. I think the $64 million question that I don't know the answer to, and I can't find it in the literature, what is the tension of DNA in a cell? And if anybody knows that, I would really love to know, because this probably is biologically relevant. I showed you data that EB47 increases binding to a nick. It also increases the binding to an undamaged nucleosome. And so some people would call this trapping on the chromatin. And as I said, we think there's competition between the nick and the binding to the nucleosome. Obviously, with cryo-EM, we'd love to get a structure of PARP bound to an undamaged nucleosome, but that's a hard problem because it's also going to form complexes on the ends of the nucleosome. Is our PARP active? That was really important in our nuclear extracts. This is a wonderful gel-based assay where you can see autoparylation if you add NAD and DNA to the extract. This is halo-tagged PARP. So the halo tag, you can run on the SDS gel and see the fluorescence. And so what you're seeing here is basically over time, it gets autoparylated and gets much bigger. And so it's very active in our extracts. Could we see single events of PARP being active in the flow cell? And that was a challenge that Matt took on. And fortunately for us, Chris Lord's lab deposited in the ad gene this, the PAR binding zinc finger domain of APLF. It's two zinc fingers that bind very tightly to PAR, and he used it in real-time analysis in cells. And we were thinking, could we use it in the C-trap? So in this experiment, we have green PBZ and red PARP with a single nucleosome. And you can see here, it takes about 400 seconds for the NAD to flow in and activate PARP. We get PARylation. We don't know if it's PARP or the nucleosome because we get landing of the PBZ domain. But then later, you can see that the PBZ domain came back. PARP then revisits the same place. We're thinking it added more PARP. And then you can see very robust binding of the PBZ domain. So we were lucky. Proteins that probably have 100 nanomolar affinities or higher, it will be very difficult to see in the C-trap, but the PBZ domain we measured to be about 34 nanomolar. So it worked very well. So because of this new approach, the guys in the lab are going full blast. And I just wanted to show you a survey of some of the proteins that we've been able to look at over the last 2.5 years. So it works incredibly well for basic excision repair and other proteins. And we're really delighted that the system is working so well for us. So I didn't have time to talk about what effects of the dark proteins, but if you're interested, I can answer that in the Q&A. So we think that SMADNE has a lot of advantages. You can rapidly look at variants. I just showed you the tip of the iceberg of what we're doing with PARP. You can look at post-translational modifications. So by overexpressing a kinase or whatever modifies your protein of interest. The chaperones was an added bonus. What we're finding, and this is very interesting, for some glycosylases, particularly 8-oxoguanine glycosylase, if you put that in the flow cell with undamaged lambda DNA, it binds non-specifically. But as soon as you take that purified OGG1 and put it into an extract, all the non-specific binding goes away. So we think there are dark proteins in the extract that are binding DNA on and off, and it shields the non-specific interaction. And these dark proteins will also then perhaps increase the dissociation rates of different proteins through a facilitated dissociation. So I think we're one step closer to understanding what's going on in the nucleus. So with that, I'll end. I've just been blessed with amazing people in my lab, wonderful collaboration with the Genome Stability Program members. We call it the DNA Pit Crew, and that's run by Patio Presco. We have an amazing imaging facility, Simon Watkins, and a lot of this work was supported by a wonderful interaction with the LUMICKS team, and I was really happy to get an S10 so we could actually buy the C-trap, and there is some other funding. So with that, I'll end and try to answer any of your questions. Thank you.
Hello, I was just wondering whether you tried the zinc finger 3 and the WGR domain of PARP1 in your binding assays.
Not yet.
But you're planning to do that?
Yeah, I mean, we're going to march through all the motifs.
Okay.
There's some evidence.
And also, do you think when you sort of separate the zinc 1 and zinc 2, so we separate from the full-length protein, like you did that, do they behave similar as in the full-length protein?
Yeah, the KD was almost identical. You could see that it's sampling the neck with almost the same kinetics as the full length.
Okay.
Thanks very much. It's very impressive how you can also get all of the rates and the equilibrium constants and the like. So you mentioned that PARP1 binds with an on rate that's almost at the diffusion limit.
Yeah.
It's kind of surprising that it would do that to a single spot on the DNA. So can you exclude that it binds elsewhere and slides to the damage, or what the mechanism is in detail?
I love the question, and thank you for that, because we've been really puzzled by that. We don't see any evidence of any sliding. So if it's sliding, we normally visualize at about 10 frames per second. So if it's binding and sliding, it's going much faster than we can see. One of the questions that we're thinking about is maybe through the non-specific interaction with the DNA, you can build up a local concentration near the DNA, and so then the orientations of those molecules would be lined up better to interact with the NIC. So imagine if it's interacting non-specifically all along. So I'm trying to think of a good way to test that idea. Yeah. I can also say that we've looked at the on rate of DNA ligase, XRCC1. They also seem to be diffusion limited. So it was a surprise to me.
Fascinating.
Okay, thank you. Thank you for the talk. It was really super interesting. I was wondering whether you see more or less the same effect in the one that is catalytically dead, PARP1, or if you use something like talazoparib, which is a very potent and trapping inhibitor.
Yeah, so the catalytically dead bound much longer. It was about an average time of 60 seconds. And so the idea is that reverse allostery, somehow it's changing the conformation of PARP so it binds tighter to the NIC. As I said, Jennifer is walking through all the inhibitors that we can get our hands on. And our first pass on olaparib, we didn't see any change in the kinetics. Obviously, it's inhibited. We can see that in our gel-based assay, but it didn't change the on and off.
Talazoparib.
No, olaparib.
Yeah,
Yeah, we're gonna look at all of them though.
Talazoparib is much stronger.
Yeah.
So maybe that you want to look at that.
We will. Maybe she has. I haven't been in touch with the lab for a couple of days.
All right, if there are no more questions, let's thank Ben one more time.